GeneSupport is a unique platform offered by Genome Biotechnologies Private Limited to provide the most customized solution towards wellness. GeneSupport is involved in studying human genes in detail to understand the correlation between our genes, diet, and fitness requirements. GeneSupport employs best-in-class sophisticated genomic research technology to carry out the detailed testing for every human DNA sample. Any individual willing to undergo such detailed genomic analysis would be counseled by our Nutrigenomic experts. The counseling would involve screening of the individual with respect to several parameters like height, weight, body mass index, and biochemical analysis. Post-counseling, the individual would be guided to take up the right genetic test amongst the various offered to suit his or her needs. The test is conducted on the DNA separated from our cells present in the mouth, for which blood is not required. A simple swab that looks like an earbud is used for sample collection. The swabs and the entire sample collection kit is handed over to the client by the Nutrigenomic counselor. The individuals can easily collect their own sample even at home by simply rubbing the swab around the cheek walls. The swab is to be rubbed against the cheek walls thrice, first in forward and reverse direction, and then up and down direction. The instructions for which are also mentioned on the kit. 3 such swabs are required for this test. Once your sample is ready and the form is filled completely, the client can inform our logistic team. The contact details of the logistic team are available on request form as well as on sample collection box. Our collection person will soon reach your home to collect the specimen. The specimen is then transported to the Central Processing Laboratory of Genome Biotechnologies located in Pune. Genome Biotechnologies is a Government of India recognized genomic R&D laboratory. The state-of-the-art lab at Genome Bio is well equipped with high-end instrumentation for genomic research and is certified by several national and international EQAS agencies like Treat Asia Bangkok and NRL Australia. The technical support desk will receive the sample and its form and log in the sample receipt in Master Data Management System and inform the extraction group to take the sample inside laboratory for the first processing stage. The member of the extraction group will carry forward the specimen to biosafety room for barcoding of the sample. Sample will be physically verified, barcoded, and then accepted at biosafety room for DNA extraction. DNA extraction from swab sample is performed in laboratory using standard procedure. This DNA is further used for analysis of multiple genes by polymerase chain reaction known as PCR. PCR group prepares the necessary PCR mix and adds the DNA sample provided by extraction group into each PCR tube as template DNA. This procedure is very critical as it involves use of PCR primer mixes for various genes to be analyzed and special chemical reagents that play vital role in DNA amplification without error. After this addition, the tubes are transferred to DNA lab, which is a high-end instrumentation lab. Tubes are properly placed in 96-well thermal cycler block and the thermal cycling program is initiated. This cycling lasts for 2.5 to 3 hours, after which tubes are taken away from the thermocycler machine. Gel electrophoresis of some amount of product is performed to check the correct gene amplification. This product is then subjected to automated DNA sequencing using a world-class gold standard DNA sequencing equipment. The DNA sequence is generated in the machine in the form of string of nucleotide bases. The quality of this data is verified by the quality manager and his team and then processed further for SNP analysis. Few genes from the panel are also accessible by using PCR RFLP methodology. Once the SNP analysis is done, the sequence data is studied and discussed by a panel including the team of experts. The mutation profile is analyzed and finalized by this panel. After this, a report is prepared by the lab team which details all the nucleotide polymorphisms in each gene and its significance with respect to diet, nutrient metabolism, and exercise. The hard copy as well as soft copy of the report is made available to the Nutrigenomic counselor for their views and remarks and then shared with the individual undergoing the test. The complete report details all the genes studied, type of polymorphism, and its significance in the individual. The Nutrigenomic counselor explains the test report, its outcome, and designs a personalized diet and fitness program for the individual. The Nutrigenomic panel offered by GeneSupport provides a detailed analysis of important genes in your body and enables your dietitians and fitness experts to design a customized program for your health and well-being. GeneSupport helps you understand what you are and thus help you live a healthy and happy life.